I see. So you've got this genetically engineered mouse. It's missing this key component in T cells of the immune system.
Mhm.
Um, and they get fat in late age and they seem to be missing certain bacteria, but then you can give them the bacteria and it reverses the obesity phenotype.
That's correct.
Hmm.
Yeah.
How do you give them the bacteria, and how is it a full reversal? How quickly does this happen? How dramatic is that reversal?
That's a good question. So we give the bacteria via an oral gavage. So it would be the equivalent of a person like just taking a pill.
Yeah, yeah, it's like taking probiotics.
That's right. Yes. And we give it to the mice every other day, so it's not a ton. The reversal is pretty dramatic. And actually, I shouldn't say the reversal. It is the prevention because we basically start giving these Clostridia to these animals before they develop
Ah, I
obesity
see. Okay.
because we knew that they would develop obesity later in life.
Yeah.
So we gave them the organism early in life.
Hmm. Hmm.
So we were asking if we could prevent the onset of obesity if we provided these organisms. So that's what we did. We still have to do the reversal experiments. We haven't done those.
Yeah, yeah. Interesting. Okay, so genetically engineered mice, they're defective in their immune system in this particular way. They
Mhm.
don't have certain microbes that they would otherwise have. They develop obesity late in life, unless you give them these bacteria earlier in life, and then they don't develop obesity.
That's right. You're really good at this, Nick.
Thank you. I'm just trying to make sure we're following. So,
Mhm.
so how do you interpret that so far? Is it just, and I may have missed this. Were you giving them back just one species, or is it sort of like a bunch of different species in this family of bacteria?
Yeah, it was a big group of
Big group.
bacteria.
Okay.
It was probably 40 to 50 different types of organisms, all of them
Hmm.
being spore formers, but all of
Hmm.
them being part of Clostridia.
Hmm. And then I assume you characterize sort of what was going on under the hood. The lipid absorption probably then just looks normal in these mice.
That's right. Yeah. When you put in those Clostridia, you resolve the increased absorption of those lipids.
Hmm. And so, one thing people might think about here that I think immediately comes to mind is, so many people have had experiences or know of people who've had experiences where, even sometimes within the same family, or, your friends or whatever, you seem to be eating more or less the same diet. It's not obvious that one person's like completely gorging, more than the other person,
Yeah.
or even people that are, really trying to lose weight and they're cutting back and they fail to do so. One person's lean, one person's not lean or obese, but they seem
Mhm.
to be eating comparable diets. This, starts to kind of sound like that, where you could eat the, it's possible. This is, I mean, I think what this shows is it's possible for two animals to eat the exact same diet and have dramatically different results in terms of body fat deposition because the microbiome is regulating how much is being absorbed.
That's right. That is exactly right. And I think it's still not a very popular thing to say because
Mm-hmm.
most people think it's, energy in, energy out. Like what you eat, if you expend the energy, you're going to be thinner. But what you just said is right. We just showed two animals they ate the exact same. They did the same movement, but one got obese and one didn't. And it was
Mm-hmm.
all based on the types of microbes they have.
Hmm.
So to me, this is a really important finding because this says the types of microbes you have are going to dictate your body composition, and this is going to dictate, health.
Mm-hmm. And one thing that's interesting, unfortunate, you might say, a little tricky, is um, these mice don't develop obesity till late in life.
Mhm.
So it's not like you could see something happening earlier on. Um, how do you start to think about, like, what that might mean for humans and sort of detecting, it's sort of like they go through their entire life and something, is different about them under the hood, but they don't develop
Yeah,
this phenotype till later in life. How do you start to think about that from more of a human perspective?
sure. So I think there's probably two things to say there. The first being um, is that we were able to induce these animals to become obese earlier in life
Mm.
if we pushed it with a high fat diet.
I see. So they more readily develop obesity. Is
That's
you
right.
almost might say that's the phenotype, and it just, if you don't push it with a high fat diet, it takes a long time to
It
set
just
in.
takes longer.
I see. I
Right.
see.
So diet was a really important component here.
Hmm.
And we can talk more about why mechanistically I think that is the case. Um, The second thing to say, I just forgot. So maybe it'll come to me in a minute.
Yeah, we'll come back to it. I
Yeah.
wanted to ask you, so when you look under the hood and you compare the mice you guys made, the genetically engineered mice that are predisposed to obesity, basically, what else, if anything, is sort of wrong with them? Do they have fatty liver disease? Are they insulin resistant? What does sort of the physiological or metabolic health profile look like?
Great question. So it's not just obesity.
Mm-hmm.
These animals develop fatty liver. They don't clear glucose as well. So they're starting to look like they're getting diabetes.
Mm-hmm. So it's basically a
Um,
metabolic syndrome.
it really is a metabolic syndrome, and I
Yeah,
think we
yeah.
use that terminology in our paper a lot. And this is, something that's becoming a huge problem in Western civilization.
Mm-hmm. And the other thing that was interesting is I think you have a result here where you took these genetically engineered mice that develop obesity. And as you said, they're missing certain populations of bacteria in the gut. They're developing metabolic syndrome, and then you just you co-housed them with other mice. What happened there?
Um, so those are interesting experiments. So this is very similar to a microbiota transfer
Mm-hmm.
study. So when you put mice into the same cage, they're coprophagic, so they essentially eat each other's poop. So that's, essentially that's what a microbiota transfer is you are feeding
Yeah.
mice.
So
Um,
by their nature, mice will automatically do a microbiome transfer.
that's right. So
Yeah.
generally, if you put mice into the same cage, their microbiome starts to converge and look more like one another. It's
I
not
see.
always the case, but for the most part, that happens.
Mm-hmm.
So when you put knockout and wild type mice together, what we saw was actually the knockout animals that were co-housed with wild types. Those animals got, oh my gosh, they got fatter. So they took on the phenotype of whoever they were, those mice that they were caged with.
Mm-hmm.
Um, those experiments are really nice because you can start to follow what microbes are moving to what animal. And that was really one of the key experiments that helped us identify this Clostridia population.
Mm-hmm. And so when you give these animals high fat diets, can you say a little bit? So, what does high fat mean exactly in terms of total fat? And is it important at all what the specific sort of fatty acid composition or the other composition of the food is?
So our high fat diet is a 45% high fat diet, which is actually low for what other people do in the field. A lot of people give 60%
Mm-hmm.
high fat diet. Most of the fat is coming from a palmitate in these diets, although not exclusively. And I think the source probably does matter, but that's
Mm-hmm.
just the diet that we.
Is that a saturated fatty acid or unsaturated?
I don't know. I think it's saturated.
Okay. I I'm
We,
I'm I'm
we
with you.
quote
I'm
me on
like
that.
80% sure it is.
Yeah.
Yeah. So it's 45% fat. I don't know the numbers off the top of my head, but I believe like what would be a typical average American or someone eating the so-called Western diet? I think it's, roughly 50-60% carbs and maybe 35-40% fat. So, I guess kind of generally in this ballpark.
Yeah, I think that's one of the reasons we used the 45% high fat diet because we felt like it was more akin to what a human might
Mm-hmm.
consume as opposed to
Mm-hmm.
60%, which just seemed outrageous.
Yeah,
But
yeah.
I guess if you're eating McDonald's all the time, you might hit that.
Mm-hmm. And so you co-house these mice. You've got the obese one and the non-obese mice. The lean mice become obese. So do their phenotypes converge? Do the obese mice become a little bit more lean, and the lean ones become a little bit more obese, or do the lean mice really just take on the obese phenotype?
Um, the lean mice take on the obese phenotype.
Hmm.
It was not a convergence, which I thought it would be.
Mm-hmm.
Um, yes.
Yeah. So, well, yeah. What
Sorry.
do you have any thoughts on like why that might be, or is that mysterious at this point?
Yeah. So I think what was happening, and again, what I said before is that these experiments were one of the critical ones to tell us that, like, help us identify who the important microbes were.
Mm-hmm.
So, because what you do is you sequence prior to the co-housing experiment,
Hmm.
and then you sequence after the co-housing experiment.
Hmm.
And what this does is it really helps you to, because the microbiome is incredibly complex. So
Yeah.
we're always asking questions about, who is it?
Hmm.
So when you do these sequencing experiments, then you can identify the key microbes. So what we did is we could find the microbes before and after transplant, and you could see the Clostridia, you could see those Clostridia moving around. And
Hmm.
wherever the Clostridia were, the mice were lean.
Hmm.
And wherever they were missing, the mice were fat.
Hmm.
So that's what really highlighted um, who was important here.
Yeah, and it's also it's kind of a convenient or like optimistic type of result in the sense that okay, you're showing that you can basically prevent a phenotype by just introducing the right bacteria. You can do this orally. Obviously, people not just in the scientific world, but out in the rest of the world, probiotics are a big thing now. There's lots of marketing around these things, but the implication is that something like this could work in principle if you take an oral pill that contains the spores of the right bacteria, at least early enough in life, that can, at least in these mice, be sufficient to prevent the onset of something like obesity.
That's right. And so we have a study that's in review right now because I told you that these Clostridia were like 40 to 50 different organisms.
Mm-hmm.
That's really hard to grow that up, put it in a pill.
Yeah, yeah.
So we've been really interested in identifying like who is the member of this consortia or members who are really driving the lean phenotype.
Mm-hmm.
Because just as you said, it seems like a really great way to just stay lean is to just make sure that you have the right bacteria.
Mm-hmm.
And we've identified one particular species in that group that's driving most of the lean phenotype.
Mm-hmm.
And we can purify that organism out and give it to mice and make them lean.
Mm-hmm.
So this is really exciting to me because now we can also go and look at the microbiomes of humans, because there's a ton of studies that characterize the microbiomes in humans.
Mm-hmm.
And if you look at humans who are obese or have type 2 diabetes, this particular species is missing.
I see.
So it really
Uh,
makes us feel like, what we're finding in mice might be applicable to humans.
I see. Yeah, I was actually going to ask that next. I think is how much does the microbiome of mice match humans? But it sounds like at least for this species, like this is we both have this in our microbiome. We can both be missing it, and there's at least a correlation between not having it and being obese in humans.
That's correct. Right.
Hmm. And so, is that like coming up next, or is someone going to test this in humans?
We are trying, yes. It's a
Yeah.
little bit trickier to do these types of things in humans because you
Right.
have to grow up the bacteria to a certain like cleanliness level.
Right. Right. Right.
Um,
And I
so
think one of the tricky things here is, well, so if we go back to the question of reversing the phenotype versus preventing the phenotype from
mm,
developing, you were able to prevent it from developing by giving mice the right bacteria, throughout their life. Um, the question is, can you, if you do the experiments where you reverse it in adulthood, that would imply that maybe something like that could work in humans. If that didn't work in the mice, then you've got a greater challenge because you've got to somehow detect that people are deficient in these bacteria early enough in life, and then supplement them with it before they ever develop obesity. How are you sort of thinking about all that?
yeah, and this goes to the thing that I forgot to say earlier because
Mm,
you asked me like, how do you parse out people who might be more prone to developing obesity or not?
yeah, yeah.
I think if we can identify the specific bacteria that are really driving the phenotype, kind of like what we're doing now,
Mm-hmm.
it's really easy to detect these bacteria, various molecular methods, PCR, for instance,
Mm-hmm.
in people's stool. And one could imagine you eventually will get to the place where people are sequencing their microbiomes, let's say, when they're younger,
Mm-hmm.
and asking, like, am I missing this microbe that will make me lean? And if so, then you could say, I'm going to supplement myself so that I can prevent late onset obesity.
Yeah.
So I do think that as we progress on in this field, that you'll be able to make these types of predictions by looking at the microbiome early in life to see what
Mm-hmm.
you have.